Good morning, friends. Myself, your botany mentor, Dr. Mukesh Kumar. Most welcome again in Hortus Botanica, where I will try my best to explore your botany under EBAJ, that is, exploring your botany A to Z. My dear friends, in last class, we started plant physiology, first chapter, that is transport in plants. Under transport in plants, we studied, you remember, the first topic, that is the diffusion. Okay, and after that we have osmosis, but before that we have also some other topics related to diffusion, right? Like diffusion pressure, type of diffusion, and we will also discuss today one question that was your homework. I'm very happy most of you replied well correctly, and again I will give you one brain scanner question as usual. So let's start first the second topic, my dear. Yeah, the second topic is your diffusion pressure. Those who joined only today, I will request you, please see the first lecture, that is your diffusion. So you will get the basic knowledge of the diffusion. So today we will discuss further in the second lecture the diffusion pressure. By name it's clear, it's the pressure, and the pressure is due to the diffusion. And in last day, remember I told you the diffusion is movement of particles, molecules of the solid, liquid or gases from the higher concentration to the lower concentration. We discuss about the second law of thermodynamics, that how the free energy is also moving from higher concentration to lower concentration. So when the particles or molecules are diffusing, then that will create a pressure, and that pressure is being exerted by the diffusing particle, my dear, and that is along with the kinetic energy, half mv square. So when you want to find out the value, then this is PV is equal to nRT, you all know, class 10th, and if you find out the pressure, then this is nR T by the V, nRT by V. So for pressure, diffusion pressure, if you want to find out, then what we have to do? Yes, you have to find out the constant, gas constant value, that is 0.831, and again the temperature Kelvin, this is 273, again it is at 10 degree, at 10, 20 degree, at 20, like that. Okay, so usually numerical type of questions are not coming here, but mighty possibility if they are asking, you can use this formula. Now let's know about diffusion pressure. Okay, what is the diffusion pressure? See, if you are talking about the pure water, see, first condition is your pure water. So in pure water, what we have? We have only solvent, mind well. So here what we don't have? Yes, we don't have any solute inside. See, this is, there is no any solute in the first case. Suppose this is your first case A, this is your second case B, and this is your third case C. So in the first case, you don't have any solute molecules, rather you have one and only the pure water, and that is your solvent. So what do you think? The molecule of the water, what happened? They have very high free energy. So this molecule will move from the higher kinetic energy. So what will happen here? They will move from the higher concentration to the lower concentration, my dear. Mind well. Okay, so if you have given water, then only water have the maximum diffusion pressure. I used to explain like that. Suppose if you are talking about the solvent, no, and then there is the solute, and both are going to constitute solution, no. So suppose the solvent is your U, or better to say your father. So what happened? The female partner of the male, that will be supposed to be the solute. Mind well. So what happened if male is there and male is supposed to the solvent and the female is going to be the solute, then their family will be supposed to be the solution. Mind well. Remember, if the male was alone without the female, so what happened? Their free energy was maximum. Or you can vice versa, I can think. My dear, you can think a female is there, okay? So the female, when she was not married to any male, then what happened? Yes, she had a very high the diffusion pressure, high free energy were there, isn't it? So what happened when they are going to the association to their another partner solute? So of course, what happened? Both will move together. So what will happen? Their my biomass will increase, my dear. Okay. So what will happen? Their kinetic energy will come down. So it was found that as there is the addition of solute, what will happen? The diffusion pressure will gradually come down. So see the first statement of what I gave here, the diffusion pressure dP maximum, where it is maximum? In pure water, which is taken as the solute here. Why? Because there is no solute. Mind well, if there is no solute, that's why they have the maximum free energy. And since they have maximum free energy, so what will happen? They will move by their maximum energy, didn't it? Now, so see in the second case what happened? I have taken here suppose 1, 2, 3 and 4. So what is being added? Solute is being added. Suppose 4 molecule of that solute is being added. Here we have more than 4. See again they have the solutes and the number is maximum. So what you are finding here, that as you are going to add here 1, 2, 3, 4, 5, 6, 7. So see here we have the how many solute? We have the 7 solutes. Here how many solutes we have? We have the 4 solutes here. Mind well. So here we are talking about 4 and 7. So gradually the number of solute is being increased. So what happened here? The dP in the first case, yes my dear, it is the maximum. But in the case of C, what happened here? The dP will be minimum. Mind well. And here in the case of B, it is in between, yes, A and B. So A have maximum dP. This is also more than B, as B is more than C, like that in this hierarchy. So as you are going to add the solute, what will happen? With addition of solute in pure water, dP, that is diffusion pressure, gradually it will decreases. So several times this statement has come in your NEET examination. Okay, now move further. We have different types of the diffusion. First we have the facilitated diffusion. So let's say about the facilitated diffusion first. So when you are talking about the facilitated diffusion means to say this is a diffusion, okay, of what? Hydro means water, philic means affinity substances. So these substances having the affinity with water, they have to move. This is the hydrophilic substances, water loving substances, they are going to move. And how they will move? They will move from the higher concentration to the lower concentration. Mind well. So if they are going from the higher concentration to the lower concentration, my dear, so what it is? It is going along concentration gradient. And remember in the last class I told you that if they are going along the concentration gradient, then what happened? We need not any ATP. So they will not consume any ATP. Isn't it? So what happened here? But what they need the carrier protein present on the cell membrane. So they need the membrane protein that is supposed to be the carrier protein that will carry the hydrophilic substances, and this is called as what? Yes, facilitated diffusion. So what is the diffusion? This is a facilitated diffusion. What should be the definition? Very simple. Movement of hydrophilic substances, yes, along the concentration gradient, that is from higher concentration to a lower concentration. concentration with the help of, yes, only carrier protein we need. We need not ATP, and this is called facilitated diffusion. Now see, in the last time I told you about the simple diffusion. Remember, with comparison, then you will retain the concept. And here we are talking about the facilitated diffusion. In facilitated diffusion, they are going from the higher concentration to the lower concentration. It was the same in the simple diffusion. What happened here? You are telling we need not ATP, not need, yes, this was same in the simple diffusion. But here you are talking that we need the water carrier protein, mind well. But for this one, what happened? We also need not the carrier protein. So here we have a basic difference along with the diffusion and the facilitated diffusion. Now you will talk, sir, why we need this carrier protein? Why we need this carrier protein? Let's get the concept. Okay, so what the concept of this one? See, what happened? You are allowing which one substances? Remember, you are allowing the hydrophilic substances, means the substances having water affinity. See, water affinity. So what's the problem here? This is the problem, that this is the difficulty level will high if you are going to transport water or water affecting substances through the cell membrane. They will create problem. Why they will create problem? Understand, because you know that we have what we have here this protein, lipid and protein we have, remember. So when you are getting this plasma membrane, you recall this is the lipoproteinous membrane, lipid bilayer membrane we have, and here we have what? Yes, this is the lipid. We have also the protein, remember, it may be the tunnel protein also, like this, passing through this one. So these are the protein. So we have both lipid as well as the protein. And you all know maximum what we have, usually maximum we have the lipid. And what is the tendency of lipid? Lipid are the substances having the hydrophobic nature, mind well. So they have got the hydrophobic nature. They are water hating, they are not water loving. So what happens if you are getting any water molecule on your cell membrane, then what happens? Cell membrane will not easily absorb it. No. Remember, take an example. If you are getting any coconut oil, suppose this is your coconut oil drop, and this coconut oil, you will know this is the oil, this is the fat, lipid. So what happens if this is your plasma membrane? Then you will find this plasma membrane easily absorb this coconut oil. Remember, when you are taking the coconut oil on your skin, see how they are very easily diffusible layer. Why? Because they have the affinity, because of the this lipid content. But water, it is what happened? Water is being repelled by which one? Yes, the cell membrane. They are not hydrophilic nature, mind well, hydrophobic they are. So what happened inside this cell? What we have? We have maximum water, 80 to 90% in a cell we have water only, mind well. So, and this water, how this is very surprising, this water is not allowing directly, but we need this water. So for that, what we have to do? We have to do some alternative arrangement. So for that, nature has created a special membrane here, my dear, and this membrane is called carrier protein or the membrane protein. What it will do? Now it will find, they are very specific in nature, mind well, they are very specific, you know, protein are specific. So it is also very specific. It will find that if something is there and like this, see, this is your the higher concentration, and same thing inside we have in the low concentration, any water loving or hydrophilic substances, and suppose this cell need this one. So what happened? This higher concentration, this is inside we have the lower concentration, mind well. But in spite of that, since their nature is the hydrophilic, that's what they are not allowing generally to go. They are not allowing. So what happened? Now this membrane protein will create, what they will create? They will create a pressure gradient, concentration gradient outside and inside first. Due to this gradient, higher and lower concentration, this membrane protein, now it will allow this molecule, because inside we need this molecule, there is the requirement of this molecule. That's why the cell will give the message to their specific carrier, membrane carrier, that please let them in, allow them to in. So this facility will be given by the carrier protein to these hydrophilic substances to get entered. So when it is required, this specific is required by the cell, so it will now get entered. Mind well. So this is the membrane protein, my dear, which provide the site for diffusion. How? Along the concentration gradient, means just I told you, they will move from the higher concentration to the lower concentration. Since they are going along the concentration gradient, so what happened? No need of pumping. So there is no need of metabolic energy, ATP. Got my child? It's clear, my dear? So see, this is the main conceptual part, that why we need this membrane protein that was not needed in the simple diffusion or the passive diffusion, isn't it? So okay, now I will show you a very good animation, so that you can get your concept very good weld. Okay, so what I told you, everything is there. One thing also, suppose you have got this periparticle, suppose this is a red color particle, but this red particle is not inside, means cell need not this particle. Mind well. So this particle will be not allowed by the carrier protein, it will be kicked out, it will be not allowed. Just I told you, because this membrane are specific, only the required things will be allowed through it. Okay, only when the site will be created, okay, along the concentration gradient. Got my child? So let's see first about that. What I told you again, this is the hydrophilic substances movement from higher concentration to lower concentration without ATP, but we need the carrier protein, that is the membrane protein, and this is called facilitated diffusion. Okay, so see what I told you, it is under a very good animation and practical. You can see this is. So see what it given here. See, this is your plasma membrane. You can understand, this is your cell membrane, plasma membrane. Got it? So I have taken here two conditions. This is your first case in green, and this is your second case in this yellow condition. See, this is nothing rather your carrier protein or the membrane protein, here as well as here. And well, so what happens here, you will find that these are the, see, this is same molecule is there. So this molecule is present in both outside and inside, and they see how they are moving through this one. See how they are moving through this one, means to say, of course there will be higher concentration here and there will be lower concentration. That's why they are moving through this one, and they see the along the concentration gradient, my dear. Is it clear now? So same thing what I told you, this is given in this diagram, no? And before that, again I will tell you about how this is going to be completed. No, let's see. See, yeah. So see, this is your first case, plasma membrane is there as I told you. Now see, this is your second case conditions, plasma membrane. See, outside what we have the more molecules, and inside what we have, see, I'm stopping. See what I told you, outside what we have, higher concentration. See, this is high concentration, and inside what we have, see, this is your low concentration. No, so what happened, since we have no any membrane protein, so see how the outer membrane where they are high concentration, in spite of that they are not allowing them inside. Why? Because they have the lipid, and lipid is not allowing them to come in. isn't it? No? So let's see further what happens here again. Yeah. So what will happen? So see the same condition what I told you. My membrane lower concentration. So outside higher concentration gradient creates. See outside again. See in the both the cases we have higher concentration outside, lower concentration inside. In the first case we need. So what we need? We need the membrane protein, and they are creating the gradient, concentration gradient. So see outside what happened? This membrane is created the requirement. Now see how it comes in. So what happened? Particle not required, so it is not allowed. It was prevented. See the same cases here also. Outside more concentration, inside less concentration. See this carrier protein is there. They see how they are allowing and blocking. Switch on and switch off is there here. Again when required, it is switch on, switch off is there, and a time will come both will get the equal concentration, and within equal concentration this is supposed to be the equilibrium state, isn't it? Yeah. Now let's see, this is the I told you about the facilitated diffusion. Now let's see about what are the other factors. So first, the size is also a very important factor. If you have got the size, smaller size as well as the larger size, this is not about the only facilitated, this is also applied for simple diffusion also. So if you have got your small size particle, their diffusion will be faster, and if you have got the larger size, their diffusion will be smaller. You can feel it. Take the sugar. If you have taken the sugar, no, and the sugar you have got this sugar small particles we have, and this is the sugar blocks we have taken. So what happened? Blocks have the larger size, so their diffusion will be what? Little slower. Slower it will be, but what happened here? Their diffusion will be very faster. So depending upon the size of the particle. Second, we have the nature of the membrane. So just I told you what happened. We have made up of both lipid as well as the protein. So what happened? Only the lipid will be easily allowed. Protein might be easily allowed also. But when you are getting water, then water is not allowed, because their transport only depends upon the that is solubility of lipid, because we have maximum concentration of lipid. So if something is lipid soluble, so what happens? They will be easily transport. Just actually before. But if we have hydrophilic, then it will be a problem. So for that problem, we need what we need, the membrane protein, what I told you. Now, so let's say this is your diagram from NCERT. Again, we are showing you the facilitated diffusion. See, this is your cell membrane. Cell membrane this is, and outside we have the higher concentration. See, this is your membrane protein we have. No, and inside we have the low concentration. So see how the higher concentration is moving through this membrane protein to lower concentration, and this is the facilitated diffusion. So first case this is. After that one, what will happen? This the particle will just rotate by this way, and as it will rotate, it will see how the particle that was present earlier here, this first particle, now this is inside first, again second is also coming. So what we found, see, this is the transported molecule that has come inside the cell. What was it, the requirement of the cell? Earlier where it were present? Yes, it were present outside of the cell. So this is the carrier protein, and due to this carrier protein or the transport protein, we got the solute from outside to the inside, isn't it? So this is the demonstration of the facilitated diffusion. Now, what are the characteristics of the diffusion? This is a facilitated diffusion. Since it is based on the protein, remember, so what will be there? Yes, they are very specific. Okay, so if protein is involved, so this diffusion is specific. First. Second, it allows cell to select substances for uptake. Remember, in the video also I showed you, if the same concentration was low concentration and high, if this was there more concentration, this was there more concentration, it was allowed through the membrane protein. But if this was there and this was not present inside, it was not allowed through this membrane protein. So what happened? For this uptake, they are very selective. Why selective? Because protein is in nature and proteins are very selective, specific. As I told you, no need of ATP. That's why this is supposed to be a type of passive process, because no need of ATP. Since ATP is not required, so we need not any pumping activity. Pumping only required if you are allowing from low to high. And since protein is involved, so what happened? They are very sensitive to the inhibitors. Mind well, Might possibly you are using say, any chemical. So chemical can stop this process. This is the chemical inhibitor. Might possibly you are using temperature. This is a physical inhibitor. High temperature can denature the protein. So the process will be stopped. So what you find here? You find here that there are some inhibitor. Might be the chemical inhibitor, physical inhibitor, might be possible any biological inhibitor too. So since protein is there, so they have inhibitor, they are very sensitive to the inhibitor, any sort of the inhibitor. Now the very important point, that is the transportation of the particle. What happened? This is the membrane and you have got that they have the membrane protein, no problem, carrier protein, and you are allowing from the higher concentration to the lower concentration, okay, no problem. But what happened? A time will come, this carrier protein is working, my dear, working. Okay? Okay? See, suppose in a temple, someone is coming in the temple or mosque, no problem. Temple or mosque. So what happened? Someone is going to serve in the temple and they are not getting any payment. Mind well, they are not paid person. So what happened? They will go and on Sunday for this service. Okay, they are going to utilize their Sunday, and morning the 8 and the evening 8, 12 hours they are serving. But what do you think? They are not getting anything, but they will do like the same way. So what happened? A time will come from morning to evening. In between that 12 about through 2 o'clock, they will get their energy down due to shifting of these particles. A time will come nearby 6 or 7 o'clock. Okay, what happened? That person will completely tired. Tired means to say they have done their maximum work and they have got their saturation. So when it is saturate, means to say what happened? This process will be now not allowed. It will be stopped for the particular some time, isn't it? So that now protein will again renature, then they will start their process. See, I have taken, suppose, see this gentleman, they are going to the complete full of energy. See how they are full of energy. So who are this? They are a very good carrier. They are carrier protein. They are going to carry so many work of the office. See, in the office they have come and they did their most of the work in the first half. Now in the second half, what happens? See, so many stress is there. Why? Because they have got their maximum work. Now they have got the attention of their saturation. They will saturate, isn't it? So really, my dear, the very good thing it is that if the carrier protein, they are going to be saturate, they will not work. Okay, so they will stop this process. This is very important because they are very specific in nature. Now, type of facilitated diffusion. See, we have, this is your cell membrane. See, this is your cell membrane, and what happened? These are the carrier protein. Okay, so suppose if you have a single A particle is there and they are moving from one direction to another direction, this is your left and this is the right hand side. So this is the right hand side. So from the left hand side to the right hand side, the single particle, they are moving independently. So this condition is called what? Uniport. Uni means single, port means transport, and transport independent. So when you have the independent movement of single type of particle through cell membrane with the carrier protein, this is called what? Uniport. But might be possible you have not single, rather you have the two type of particles, C, A and B. And here again we need the carrier protein. And they are moving in the same direction. See, they are moving in the same direction. So if they are moving in the same direction, then such transport is called same transport. Okay, so here again might be possible the direction is changed. See, A is going inside but B is coming outside. So again we have got the two molecule movement but in the opposite direction. So you can say here the second and third condition is not uniport, rather this is co-transport. This is the co-transport. Two molecules are moving either in the same direction, then it is same port, either in the opposite direction, then this is called as the antiport, depending upon the requirement of the cell. So how many types we have? Three types: uniport, same port and antiport. In all the things what we need the carrier protein, but we need not ATP as they are the facilitated diffusion. Now the next one, my dear, that is the active transport. See, again, a very unique things we have. Active transport. So many times we understand, I told you that if you have a membrane, they are the high concentration to the low concentration. If they are going, this is your diffusion or simple diffusion. But you are telling you have the high concentration, low concentration due to the nature of this molecule. They are the hydrophilic, so we need the membrane, and if they are moving in the same way, then you are telling this is a facilitated diffusion. Okay, so two type we understand, we studied, my dear. Now see the third condition. Might be possible outside we have the low concentration and inside we have the high concentration. Okay, and what happened, they can't move independently, so we need here the same carrier protein, mind well. But carrier protein was also using earlier, but here what was the new cases, that was the high to low, so they are moving along the concentration gradient, mind well. Here we are finding that they are not along, rather they are moving from low to high. So they are moving, how they are moving? They are moving against the concentration gradient, my dear. See. So if you are allowing against the concentration gradient, then what happened? We need pumping. We have to pump it to do this special process, and for this pumping we need the energy, and that energy is the metabolic energy, ATP. So ATP will be involved, and that's why this is called active transport, because if ATP is consumed, then this process is called what? Active transport. So what is active transport? This is the diffusion of substances from lower concentration to higher concentration. So this is the against the concentration gradient, and for this against we need the pumping. For pumping we need energy in terms of ATP, and this is called active transportations, active transport, isn't it? Yeah. So let's see now second, what are the main characteristics for the active transport. No, first same thing here. Also we need the carrier protein. I told you, no, here you have what you are doing, allowing the low concentration to high concentration. So what you need? You need the carrier protein. First, carrier protein is involved. Second, energy is consumed. Why? Because there is the pumping activity, is involved. Okay, second, they are going against the concentration gradient, as I told you. So since protein is involved, carrier protein is involved, so what happens? They will get their saturation when they are going to attain their maximum work. No, so at maximum what they will achieve? They will achieve their saturation. No, and yes, if something is moving from the lower concentration to the higher concentration, by the use of what? Yes, by the use of ATP, okay, that will convert into ADP. So we have consumed our phosphate energy. So such types of the movement from lower to the higher concentration, this is called uphill movement. Uphill means to say pumping is required from the lower concentration to the higher concentration. So you can say if someone is going from the high to low concentration, this is called downhill movement, and for the lower concentration to higher concentration we use the term what? Uphill reaction. Okay, now yes, my dear, again I will show you how this is occurring, the active transport. Okay, so see, first of all, in active transport, how they are occurring, my child. See, so this is the diagram. Again I told you this is a cell membrane. No, this is your cell membrane, and here this is your carrier protein, and see, this is the ATP molecule. This is the energy, created energy. Why? Because they are moving from the lower concentration to the higher concentration, and this is what? This is the movement of the lower to the higher concentration. So this is the, yes, against the concentration movement. So see how it is going to be completed. No, see, so see how protein is coming. See how the protein is coming, and you feel that they are going to charge, and according to that charging, see, Huh? Color is there, protein is there, and this protein is going to charge them by y. So see what I told you, how this is the energy, consume energy. ATP is there, once it will pump, after pumping this ATP will convert into ADP. Again we need another ATP so that this carrier protein can be charged, and once it will charge, then only it will allow the movement from the lower concentration to the higher concentration, my dear. Okay, so just I told you the same way, no? So see again, see how they are pumping and are allowing the molecule. This is the energy as I told you earlier also, and they are pumping and allowing from the low concentration to the high concentration. Now let's see what are the main comparisons. If you will compare all the three what you have studied, that is a simple, facilitated, and the active diffusion. So what are the main differences? See, membrane protein. What happened? We don't need. I told you. But here, yes, we need it. We need it. When you're talking about the highly selective nature, this is only possible selection if they have got the proteinous nature. Mind well. So there is no protein, that's why there is no selection is there. Protein was present, that's why there was selection. Protein was present, so there is selection. Simple. Now if how they are allowing the movement, you will found about the movement, I will add it, then it will be more easy to understand. So if you are talking about the movement, so here movement is what? This is your higher concentration to the lower concentration, higher concentration to lower concentration. In this case there is again higher concentration to the lower concentration. In this case opposite, lower concentration to higher concentration. So low to high is there, that's why we need, what we need here, uphill movement. Here we need not uphill movement, here we need not the uphill movement. And for uphill movement what is required? Pumping is required. For pumping what is required? ATP is required. So same, see uphill movement, yes, not required, here also not required, and here it is required. So what happened here? You'll find since they are protein, so ATP, see, not consume, not consume, this is consume. Why consume? Because only you have this one, the uphill reaction. Second, since you are using the protein in facilitated and the active, so protein are highly sensitive. See, no protein was used, so they are not sensitive, but here they are sensitive, here they are sensitive, my child. No, and again, since the carrier protein is involved and they will get their saturation. So saturation will be achieved by the facilitated diffusion as well as the active diffusion. It will not achieve by the passive one. Mind well. So these are the main 1, 2, 3, 4, 5, 6 and 7. These are the 7 important differences among what? Yes, simple diffusion, facilitative diffusion and the active diffusion. Is it, my child? Okay, easy? Yeah, so I think you got the all the basic idea. Yes, my dear. Now I'm very happy you replied well. The first lecture question, that was a brain scanner. What was the question? Question stated that you have to tell the odd one. Odd means to say you have to tell about the incorrect statement with respect to the diffusion. So okay, come. Diffusion is a slow process, correct? They are not independent of living cell. Remember I told you that what are they are? They are independent. Independent. They are talking not independent. Not independent means to say what? It is dependent. And if it is dependent means to say they are using ATP. This is the meaning of this sentence. But you know that is simple diffusion need not this ATP. Mind well. So what are they? They are not dependent, rather independent. So this statement is wrong, my dear. Now, so this is, I told you, ATP is not consumed. So they are what? They are the passive process. But here what is written? Not passive. So this statement is also wrong. Now it is obvious, very clear in gas, liquid rather than solid. 100% correct. This is your NCERT line also, we discussed last class. And this is the only method, yes, only method of gaseous exchange, only in plant. Mind well, not in the animal. Animal have their circulatory system, they have the respiratory system for the gaseous exchanges. Mind well. So this is not for What? Only for this one? No, for animal we have so many other method also, but for plant it is only correct. So this statement also goes wrong. So how many statement is correct here? C, only your C is correct. So C only, yes, correct. A, B, C not correct. A and D not correct. All except A. No, we have only the C. So this is also not correct. So your correct answer is yes, A1. So very congratulations all those who replied your correct answer for this very conceptual question. Now my dear, at the end again a homework for you. Yes, again I will discuss this question next Sunday. So your homework is the new brain scanner question. Yes, this one. This is your brain scanner. You have to find out on the basis of the different characteristics what should be the correct answer. Okay, yes, you will answer your answer this question in the comment box also. You will also please like this and give your comments what is your feeling. Accordingly I will try to serve you better. Okay, so thank you my dear. Have a nice day. Meeting you again next Sunday with the next lecture. That should be the third lecture, and that will enrich your knowledge under Hortus Botanica EBAJ. Have a nice day. Take care. This is the COVID period. You all know Omicron are spreading daily. So yes, be safe yourself. You all know the three important process. Use the mask and do the good duri and wash your hand and face regularly. Okay, so all the best. Have a nice day. Take care. Enrich your knowledge of botany and be the very ideal one. What I given, identify your real grace of your face through spiritual view in the same mirror as your DNA is unique. You are unique. So please identify yourself. With these good words, have a nice day.